today to introduce the first speaker of this webinar, Dr. David Thomas, who's a professor of medicine and epidemiology at Johns Hopkins School of Medicine. David and I kind of grew up in the heydays of hepatitis C when it was first discovered by our distinguished speaker tomorrow, Michael Houghton. And I think David and I kind of really went through all the great advances. We're very happy to be part of this process. And David also over the year has made seminal contribution to the genetics of hepatitis C. And so today he's actually going to tell us about the urgency in terms of developing hepatitis C vaccine so we can effectively control and hopefully eliminate hepatitis C. David, please.
Great, thanks a lot, Jake. And thank you, Raj and John. Sorry, I can't do two things at once. Let me just get this going. Okay, yeah, thanks a lot for that introduction, and thank you for your leadership in this important, really nationally and globally in efforts to eliminate hepatitis C. So I'm delighted to kick off the webinar. My charge is really pretty straightforward. I'm going to have two things that I'm going to say over and over again, and that is that a vaccine is urgently needed and that a vaccine is possible. Urgently needed and possible. Well, unfortunately, the first bit has been evident since the mid-1995s. It really wasn't long after Michael had discovered the virus that we knew that most persons who injected drugs already had hepatitis C virus infection. These are data from the ALIVE cohort, which was a group of persons who inject drugs that were recruited in 1989 by doctors Vlahoff, Polk, and Nelson to really study the natural history of HIV and other infectious diseases. And in more than almost 3,000 persons who injected drugs, even in 89 when they were enrolled, 89% already had antibodies indicating that they had hepatitis C virus infection. And even amongst those that initially were uninfected, a third were infected in the ensuing couple years. There were the predicted relationships between the intensity and duration of drug use. And if you honed in on, for example, subsets of those who had used for 3 or more years, of persons who were HIV co-infected, nearly all, I mean, you know, plus 95% had evidence of hepatitis C. So the first bit that a vaccine is urgently needed was evident that far back. Now, of course, the vaccine doesn't help those that are already infected. These persons will have to wait for 20 to 25 years for the direct-acting antivirals, a point I'm going to come back to at the very end. But nonetheless, it's evident that the enormous burden of infection in persons who inject drugs underscores the need for a vaccine. So vaccine urgency evident right from the beginning. Now, what about the possibility of a vaccine? Because that was sort of, as Jake said, you know, back then, not everything that's obvious now looking back was so clear. And whether or not it would be possible was really an important question. One of the first clues came from looking at these individuals with acute hepatitis C infection and watching what happened over time. There were two clear outcomes as shown by the summary viral loads of the two groups on the y-axis in this figure. You can see that the majority had initial detection of viremia, just as you have become familiar with, and the virus was there every other time that you looked. But then another group, a subset of in this instance, only 15%, had initial viremia, in some instances indistinguishable from the other group, but then viral clearance and no more detectable viremia over time. And in addition, in data that I'm not showing, no virus could be found in their livers. And most importantly, there was no evidence of liver inflammation in this group, so no evidence of liver disease. Acute infection, but then successful immunity without any ensuing liver disease. So clue number one, that there might be a possibility of forming a vaccine, really came from these types of observations of successful natural immunity. In truth, the data from Harvey Alter and others and transfusion recipients had already shown this, had already shown two patterns of viremia, and that some successfully cleared. The drug use cohorts allowed us to ask a different question and to say, we knew already that unfortunately most persons who inject drugs keep injecting drugs. So if we took a little closer look at the individuals in whom we detected no subsequent viremia, we not surprisingly found that that wasn't because they stopped using. There still was a lot of exposure to hepatitis C, just as much as when they had initially become infected, but yet no subsequent viremias. And I remember talking to Bob Purcell, Jens Buch, and others, and Michael Hound about this back then. And we all thought, wow, this might be a clue that not only could successful immunity be achieved, but it might even protect against reinfection. To quantify that and to get at that hypothesis, Shruti Mehta led a series of studies where we compared those individuals who were initially infected, 164 people never infected before, who got infected for the first time. And then after testing thousands of specimens from all those that were antibody positive in this cohort, we were also able to pull out 98 individuals where we were rock solidly sure that they had had clearance for 2 years, actually. So no late clearance or anything like that. These people had clearance. The other people had never been infected before. And we reasoned that 3 things would be possible if there was protected, or 3 things would be evident if there were protective immunity. First of all, we thought that there should be fewer instances where we find viremia. And indeed, although it wasn't zero in this larger group of 95 compared to the 6 that initially were tantalizing, there were half as many viremias detected after adjusting for drug use frequency and all the other things that we could think about to adjust for. There were still fewer viremias detected in the group that had had a prior successful clearance. Secondly, we thought if there was a viremia, it would have to be of lower magnitude as evidence that there was some immune pressure early on the virus. And indeed, the peak viral loads, the highest that we detected in these individuals who had had prior infection and then another viremia, was 2.5 logs lower than in the individuals infected for their first time. So fewer viremias, and when they did occur, they were of lower magnitude. And then thirdly, and most importantly, we thought those viremias should persist less. They should be cleared more if indeed this is some form of protective immunity. And although the initial viremias in those infected for the first time, 84% were persistent, in this group, only 33% were persistent. Evidence that there was some additional benefit protecting against virus persistence, and really raising the question of whether or not similar immunity could be achieved with a vaccine that would protect, perhaps not be sterilizing, but perhaps protect against virus persistence and liver disease. Well, Andrea, who was involved during that study, also took this a step further. And Andrea said, okay, well, now let's switch to the BBASH cohort where we study people that every month, and let's ask if we can compare the same person's viremia, not a group viremia, but let's compare what happens in the same person if they're reinfected to their initial infection. And sure enough, just as she predicted, the duration, how long somebody was viremic, was longer initially than the second time. And the peak HCV RNA level shown on the right was also higher in the initial infection than in subsequent infections. All of this together suggesting in cohorts of persons who inject drugs that some form of protective immunity might be possible, at least from natural infection. Now, evidence that immunity could be achieved was sort of complemented by evidence that it could be lost. All right, and this one requires a little bit more thought. But in our cohort, persons who injected drugs who were HIV positive were far less likely to have viral clearance than those with persistent infection. Okay, so that's reasonable. They're immunosuppressed. So why is that support this concept of lost immunity? Well, in Baltimore, most of all these infections in 1989, Hep C infections, antedated the HIV infections by a decade or more, because HIV didn't even come in to Baltimore until the late 80s and early 90s. So to a large extent. So at a sort of epidemiologic level, this suggested that these people would have already cleared their Hep C, been moving through time with that protective immunity, and then after getting HIV would have lost it and become therefore that associated with persistence. And that's kind of, I didn't put that too well when I just said it. Maybe one specific example will help. Here's a single individual to illustrate. Notice in the top frame on the y-axis is the HCV virus load. And you can see a typical primary infection, viremia detected and cleared. So viral infection with clearance. Then if you drop all the way down to the lowest graph, you can see a summary figure of the drug use practices, which continued all through time. And despite that, the person had no more viremias for years. But then in the meantime, they acquired HIV in the mid-90s and then had subsequent CD4 decline. And then eventually, if you look to the far right, back up in the top again, you'll see a viremia again that persisted. So in this instance, it's an example of immunity that was achieved and then lost by HIV. And it really right about the same time, Chris Walker had experimentally sort of interdicted cellular immunity experimentally and pharmacologically in the chimpanzee model and shown the same thing essentially. And so it's really evidence, I think, cumulatively that some forms of protective immunity can be acquired. And then the complement is that they can be lost. Now, there were lots of important studies done in the chimpanzee model that I won't have time to go over in detail. Jens Buch, Bob Purcell, Marion, many of those, Michael Houghton's group, I won't show, but just a couple of points. First of all, really early on, Patricia Farsi and Bob Purcell had shown these sobering data from an animal that had been given an inoculum F. See the F up there, so followed by a typical infection with viremia that cleared in the solid lines, just showing the PCR positivity, and an ALT elevation that was sort of in the 200, peaked in the 200s and then back down again. And then autologous inoculation, the same inoculum back again. And yet, despite the fact that they had cleared that inoculum once, this animal was infected again with viremia and with a slight increase in ALT. Now, at the time, that kind of sent shockwaves through the field, and everyone's like, oh, well, we're never going to have a vaccine. Don't work on that. But notice the viremia is of shorter duration. And notice the ALT is of lower magnitude. So there is some immunity. It's a matter of half empty, half full kind of interpretation of these important, very early data. Likewise, I'm going to show these data that Chris developed because Amy Weiner and Chris, when using a Chiron, showed this important study, which was kind of scary when it came out, which is evidence that the immunity that could sort of clear or control an infection and dominate a quasi-species could be abrogated by a single amino acid substitution in a CTL epitope. And so So if you look at the top, you'll see that there's the inoculum, and this is one of these sequence alignments. So these are the amino acids of the inoculum, and then how subsequent virus amino acid sequences compare, and the epitope is in the block there if you're not used to looking at these things. And you can see that there's this substitution for the glutamate that is evident and occurs in conjunction with a substitution in the quasi-species of this virus as the principal sequence. And of course, they did elegant immunology to show that that also abrogated the CTL responses. And so without getting into too much detail on the immunology of this, a single amino acid substitution being enough to kind of shift the fitness of the quasi-species and select for escape mutation like this also was threatening to the notion that with this virus, where viruses could be 30% different in their amino acid sequence, one from another, how would we control, how would we do this with a vaccine if a single amino acid substitution was sufficient to abrogate it? And I think that is an important question. I know others will tunnel down on that more in the talks to follow. But to investigate that further, Stuart Ray and Robert Lamford did an important study where the reasoning here was if that kind of if that kind of single amino acid mutation was sufficient to lose that protective immunity, then probably if you gave an inoculum from a different genotype, then it should persist. There should be almost no evidence of any protection. So in a series of studies where persons who were animals initially infected with genotype 1 were rechallenged with a different inoculum of 10 to the 6 viruses of each of the inocula from a different genotype. In this case, on the slide, I'm showing a 4 that followed a 1, and then a mix of 1, 2, 3, and 4 all together in an animal with 1. The initial infection shown in the light gray, the secondary infection shown in the dark, with a virus load on the y-axis again. And so you can see that there's evidence in these animals of cross-genotype protection, that this sort of protective immunity that I've been alluding to and developing the case for can even cross between the genotypes. And Stewart's famous for saying that a genotype isn't a serotype, and making this point that the types of immunity that we're used to describing and highlighting this paradox that a single amino acid is enough to abrogate a CTL response, or in work you'll see later, antibody responses, is also paradoxically that this kind of protective immunity can be achieved. These animals all had high, almost immediate ISG responses in their liver, and the inoculums weren't flawed. We also infected a previously uninfected animal and got a typical primary infection just to prove that we weren't using inocula that were wrong. So I think that this is an important counterbalance to the notion that this will never be possible, because at some level there is cross genotype protection. So I'm going to leave it to others to understand the immunology better, to describe this paradox in more detail. I will say that to understand that better myself, I pulled Chris's paper out, and I found it interesting that amino acid mutation that he had was actually conserved across genotypes. So even that was not a genotype specific escape mutation. It was nonetheless. So I'm looking forward to learning from the immunologist how to interpret this, what the measures are going to be, and how we can harness this information and make vaccines that will deliver the same sorts of protective immunity. But now I'm going to come, as I promised, back and finish with readdressing the urgency. You thought I was done with urgency, and I think I really was. I think a vaccine is urgent, but some people are going to say, "Are you sure?" Because you know, in the 90s, in case you haven't been paying attention, we now have these DAAs that can cure the infection. And why do we need a vaccine if we can just cure it real easy and just, you know, eliminate infection that way? And so I think that's a reasonable question. I'm going to finish with that. So let's come back to the lab cohort. And this is work that's been led by Javier Cepeda. Now looking at 1,289 HCV RNA positive. These are people that are still have persistent infections, still alive, still in the cohort by the 2006, and started having fiber scans done to measure cirrhosis every 6 months. And you can see that the rate of cirrhosis shown in the blue bars in the y-axis is rising in the cohort, steadily rising. Just as one would predict. And then the proportion with viremia shown in the orange staying high on the Y2 axis there. But then the DAAs come, and even in Baltimore, and even in persons who inject drugs with limited access and all the stigma and all the barriers, we see by the end of 2019, almost half of the cohort having virus clearance, and the rising arc of cirrhosis being bent. Okay, so yes, DAAs are fantastic. They're fantastic for people who inject drugs, and they can reduce liver disease and help set the stage for eliminating hepatitis C in persons who inject drugs and who have already been infected. But back to the urgency question, in the meantime, the same 5 years, the incidence of hepatitis C has risen and almost doubled in the United States and in Baltimore. So even though DAAs have been out there, and even though they're being used by some, unfortunately not all persons who inject drugs, the incidence of hepatitis C, the point of the vaccine to stop incidence, that continues to increase, rising from an estimated 33,000 to over 50,000 by 2019, and that's still going up, and we all know it. So as Dr. Fauci said, because of the expanding opioid epidemic. So is a vaccine necessary and urgent? Yes. Is a vaccine possible? Also, yes. Are there challenges? Yes, again. And I'm looking forward over the next 2 days to think about that with greater detail. But no matter what, we're certainly going to need to resource that effort to achieve that outcome. So thanks very much for your attention. I used a lot of people's data and I tried to recognize them as I went along. Thank you for your attention.
Okay, thank you very much, David, for that wonderful presentation. Let's see if anybody have any questions. We can take a couple of questions. You can post it on the chat, and I can read it to the audience. Let me see. Guess anybody? I don't see anybody in the chat. So David, so let me ask you a question since I don't see anybody. So I guess a question, your group as well, many groups actually has shown the importance of genetics in terms of viral clearance, right? So do you think kind of using genetics we can harness some information from that for vaccine development and perhaps somehow trying to stratify kind of if you have certain favorable genetics, maybe certain vaccine will work well or better, or from that point of view, I know it's kind of a loaded question, but I'm just kind of asking you whether you think that something could play a role in terms of vaccine development.
Yeah, thanks a lot, Jake. I will address that. And also in the Q&A, there's 2 questions, not in the chat.
Oh, okay, Q&A. Okay,
no, it's okay. Chat. I can see them, and
okay, go
I'm
ahead.
happy
Yeah,
to field those as well. So yeah, I mean, we had hoped that by understanding successful immunity from all perspectives that we would inform a vaccine. Andrea has led the immunology, certainly the cellular immunology. Justin and Stuart have been investigating humoral immunity, and you'll see their data. And then we have, as you said, looked at the underlying host genetics as well, sort of to try to build a model of successful immunity. And I will say, yeah, I mean, interferon lambda is extremely important and explains about half of the difference we see in persons of African ancestry and their propensity for virus persistence. And we also have some pretty strong alleles from HLA that, you know, might actually be informative. But to date, I will say that's actually, I will acknowledge, has not leaped out, has been has not yet translated into a obvious vaccine solution, but we're still hopeful that it will. Obviously, the Lambda discovery contributed to treatment and to even diagnostics, and made it to all the way to human use for that. So we're excited about the findings. Stuart asked about, to just point out, Stuart Ray, the inocula that was used in this studies I mentioned that show cross-genotype protection that he and Robert did were 10 to the 6th genomes from humans. So these are far above the chimpanzee infecting dose that Jens had so carefully worked out with Bob in the 90s. Nida Ali says, what about waning immunity? That's a good question. After, you know, we're all interested in whether we need a 4th booster dose, you know, so of COVID vaccine. So this notion of immunity waning is certainly important. We're confident that it wanes when you get HIV infection and a sort of a CD4 knockout, if you will. Whether or not time itself does it, it is probably true, but I don't know that to be true. And maybe Andrea or somebody else wants to show actual evidence of it. I would guess it's true, but I don't know that. A guy named Jay Hufnagel says, can you mention whether or not, hi Jay, by the way, can you mention whether there were different rates of chronicity from acute by sex and race? So that's a great question that I did not get into. And yes, Dr. Hufnagel is right. Women were about twice as likely to clear infection as men. A study that, a finding that we actually had a hard time to disentangle from age in our cohort, but later in the INC3 cohort, Kathleen Page made it crystal clear that that was in fact, it was a woman, that it was the sex, biologic sex that did that. And to date, we don't understand why. We've looked at the sex chromosomes for obvious differences, but have not found a sex-specific basis for that. And then persons of African ancestry were more likely to have persistent infection, not clearance. And half of that was explained by the discovery of the lambda interferons as being different, their genetics being different in persons of African ancestry. And in particular, they're actually producing lambda 4 in comparison to persons of Asian or Caucasian. Let's see.
Okay.